Assalamu alaikum students. This is Tabassum Rahman Sanfi, lecturer of Biotechnology program. Welcome to my course Microbial World, which is BTE 102. Well, this might be our very first meet and we can be introduced to each other, or I can be introduced with all my students individually. However, due to the pandemic of COVID-19, this time we have to start the class at a different way. We have to go online and we have to record all the lectures in order to proceed the whole semester. Anyways, I hope all of you are in good health. So before jumping to my first lecture, I would like to introduce the course content with you all. As you have already known the contents or you have had some little idea about the course materials. This is the introductory microbiology course where we will be introduced, or you will get to know about different microbiological agents, their functions, how to grow them, and their impacts in our day-to-day life. So the course is designed in such way where I will give you an introduction or a brief history regarding microbiology, how the term microbiology has been evolved, and how the basic sector of microbiology has established in such way that today a major portion of the biological science is contributed by microbiology. Then we'll learn details about the microorganisms such as virus, which is a threat for this present era. You know about the SARS-CoV-2, which is causing the disease of COVID, and due to this it has been named COVID-19, which is SARS coronavirus disease, and it has been spread from 2019. That is why the name was designed in a way like COVID-19. Whatever, we learned something about virus and SARS-CoV-2, of course, and some other viruses, bacteria, parasites, fungi, algae, and protozoa. Then we'll learn about some infectious diseases, their pathogenic interactions, their mechanisms, and symptoms and cure. So far, this is my design, or this is my aim to let you know or to deliver lecture regarding this. Now let's start with the history. This is the first lecture. You can see the title of the lecture in the screen, which is Introduction to Microbiology. The first question: what is microbiology and why we need this sector? Like, there are several other sectors in biological science, which is biochemistry, molecular biology, genetic engineering, which is a very recent subject, but why microbiology is required, right? And what is microbiology? Now let's see, the answer is given here. Life as we know it would not exist without microorganisms, also called microbes. In lecture 2 or in lecture 3, you will get to know that microorganisms or prokaryotes are the first source of life on earth. They prevalent on earth before us. That means they are ancient than us. That is why it has been said that life as we know it would not exist without microorganisms. And why microbiology is the study of, so that microbiology is the study of microorganism. To study about microorganism, microbes, their functions, properties, anatomy, genetic constructions, the genetic modifications, everything is in the zone of microbiology. So let's see why we should study microbiology. To understand how microbe affects human health, the first and foremost concern, or the first and foremost reason that provoke us or provoke human to study about microorganisms. Otherwise, no one would bother about studying microbes or a little invisible particles or microscopic particles. All right, they have a huge impact on human health. That is why people are eager, or people have to learn about them. People have to know about them in order to keep themselves safe. So this is the first reason, which is to understand microbes, how microbes affect human health. The second point is understand life process of microbes that are common to all organisms. To understand how they are surviving, what is the survival energy for them, what is their mechanism or inner regulations. Everything should be learned or known in order to investigate about any particular microorganism. And the third one is understand how microbes can be used for our benefit. Biotechnology, which is application of biotechnology. Biotechnology means applying biological entity in order to have a technological purpose, or in order to have a technological approach. And these biological entities are mostly microorganisms, either a bacteria, fungus, or any other microbe. Alright, so these 3 points are very important. Now let's see the history. Who are the scientists or who are the warrior behind this firm establishment of microbiology? Here you can see the name of a very famous scientist, which is Antony van Leeuwenhoek. The name is a bit tough or a bit difficult too, but I think all of you know him. Who is Antony van Leeuwenhoek? He once used to work in a shop. Alright, he once used to work in a shop in order to investigate the fabric's nature, whether that fabrics would be strong, or whether that's loosen or have good quality or not. He was responsible to study about the fabrics, whether or the quality of the fabrics. However, now he has become a famous microbiologist only because of his curiosity. Let's see how. So Leeuwenhoek, the man who first discovered and documented the microbial world, he has been praised for his first documentation regarding microbial world. Leeuwenhoek invented the simple microscope. He himself invented a simple microscope from his extreme interest or extreme enthusiasm to know about the invisible particles, because his work was kind of knowing or judging the invisible little thread structure of a fabric. From this, he has developed the enthusiasm to see all the invisible particles, all the microscopic particles. That is why he first invented his own simple microscope, and this was in the period of 1673, which is almost 400 years ago. Observed the major types of microorganisms. He referred to them as animalcules. He was not conscious about the microbes, or these are called microorganisms. He just know that these are some animal-like particles and they are very small, like molecules, small molecules. That is why he named them animalcules. Well, there is a spelling mistake. I'll correct it later, but there should be an extra L. Please keep in mind it should be animalcules. Alright, not animalcules. I'll correct it in my side. So, microbes are organisms that are generally smaller than can be seen with unaided eye. That is why he had invented his own microscope. Then there comes the next scientist who also contributed a lot by identifying or discovering how the world or how the microorganisms can be classified. Because now we know that there is a huge amount of microorganisms present in this universe. If we cannot classify them, or if we cannot organize them properly, it is impossible to learn about them or to store a data about them. Alright. So the scientist named Carolus Linnaeus developed a system of general classification. He first developed how the organisms can be classifications, what should be the aspects, what should be the factors or class in which all the microbes will be dumped, or all the microbes will be sorted orderly. So microbial cells can be either prokaryotic types or eukaryotic types, but there is another information. You can see the field of taxonomy he initiated. So if I ask you the question, who initiated the field of taxonomy, you must answer it is Carolus Linnaeus. And let me make a thing clear for you all. In my course, I will always give emphasis on the discovery of scientist, not the date or the period. Alright, you don't have to go through or memorize all the dates, like in which month or even in which year the Carolus Linnaeus did this, or Leeuwenhoek invented his microscope. You don't need to, it's up to you whether you want to memorize that particular period or not, but you must go through the discoveries and you have to know that this scientist is responsible for this particular invention. Alright, the discovery is very, very important. For example, initiating the field of taxonomy is referred to the scientist Carolus Linnaeus. Now let's see what is microbial cells can be. Microbial cells can be either prokaryotic types, which is without any nucleus or without any true nucleus, or membrane-bound organelles. Another is eukaryotic type cells having true, properly developed nucleus and having some membrane-bound organelles. There are 6 main classes of microbes that we will discuss. Well, I think all of you have a bit of idea about this. The first one is fungi, protozoa, algae, prokaryotes, that means bacteria and archaea. Remember, in prokaryotes there are only two types of microorganisms. One is bacteria, another is archaea, which is almost similar like bacteria with having slight differences. Helminths or parasitic worms, and then viruses, the most important particles for this 2020 or for this year recently, because recently we have seen too many virus attacks: Ebola, SARS, MERS, then so COVID-19, Nipah virus, everything, every deadly disease, mostly they have originated or caused by a virus. And another important information is those viruses are mostly zoonotic. All right. This word might be new to all of you, but zoonotic means the viruses that have transmitted to human from an animal source. Like you know that SARS-CoV-2 have been transmitted to human from bat. All right. People ingested a bat, and from that source, these viruses have come to human. So these are called zoonotic viruses. So these are the classifications that we will learn gradually in our discourse. The golden age of microbiology. Well, an explosion of interest in microbiology centered on four main questions. How people become interested in microbiology? The answer evolves within four different types of questions, the answer to which provide a framework leading to the modern age of microbiology. Well, what are these questions? Is spontaneous generation of microbial life possible? The first thing that arises within people or within scientists is that whether a microorganism can be generated from a non-living things, like from a wooden particle, from a steel, from a stone, whether they can be evolved, originated from a non-living thing or not. This was the first outcome of their curiosity, the first type of question. We'll see the results or the answer of this curiosity later on this lecture. The second question is, what causes fermentation? People know about fermentation from the ancient period. They used to make bread with yeast, by yeast, or they used to create wine from grape and other type of fermentation products. But how this has been possible? All right, this was the second question. What causes disease? The third and most important question: how people get infected with having some different symptoms, like having cold or having runny nose or severe aches, whatever it is. So this leads to the third curiosity, that how disease has been generated within a human. And the final question, which is how can we prevent infection and disease? So these four questions arise microbiology, or these four questions has forced scientists in order to go deeper in the world of microorganism or world of microbiology. So spontaneous generation. The answer of the first question: is spontaneous generation of microbial life possible? Regarding this, there have been two different types of theory arise. The first one is biogenesis theory, which say that life arises from pre-existing life. That means to arise a new life, there should be a pre-existing life. And opposite to this biogenesis theory, which is spontaneous generation theory or abiogenesis theory, where it has been claimed that life arises from non-life or non-living particle. And there was a huge debate regarding these two theories in order to establish the proper one. All right, we'll see the debate in our next portion. Thank you very much.